Okay, this session, which is for advanced cell biology, is on apoptosis, autophagy and necrosis, and we're going to be studying different mechanisms of cell death. So after this session, you should be able to describe the different forms of cell death that occur, particularly apoptotic cell death, and contrast this with necrosis, and distinguish situations where apoptosis occurs, where autophagy occurs, and where necrosis occurs. And over the course of the next few lectures, you should be fully aware of the differences between necrosis and apoptosis, how these relate to tissue homeostasis, and we will cover in future lectures how to tell the difference between them with biochemical tests. And we'll touch on how growth factor signaling, which traditionally you consider to be promotion of growth, will also drive survival and prevent apoptosis from occurring. So there are two main forms of cell death, and that is necrosis, which is just cell death, and programmed cell death. And within programmed cell death, apoptosis is by far the most important and widely studied mechanism. So programmed cell death is just a term that describes cell death that occurs in a predictable manner, usually in response to some sort of signaling. And almost all programmed cell death is apoptotic, although other forms of cell death, particularly ferroptosis and pyroptosis, are becoming more well studied. As far as you're concerned at this level, apoptosis and programmed cell death are effectively the same thing. And what we can see here, this is an image of a mouse's paw showing the formation of digits, and you can see in between each digit there are these black cells, and they're black because they've been stained up for the presence of apoptosis. And apoptosis is shaping the tissue structures here, so it's causing programmed cell death of the cells between the digits to allow those digits to form. So this is a crucial process that helps to shape organisms. So as organisms grow, you can have periods of growth and then specific areas that die back. And this is just illustrated in a slightly different way with a different stain, again using a mouse's paw. Now in adult tissues, tissue architecture is mainly maintained by apoptosis, so where death is required, it tends to occur due to apoptosis, and apoptosis is an active process. In contrast, necrosis is a pathological response to injury, and this can be anything, some sort of physical or chemical trauma. Hypothermia, frostbite will cause necrosis. Basically, they will cause the cells to lyse and burst open, as does physical heat and chemical trauma. Ischemia, or lack of oxygen to tissues, can be seen here. So here this is an example of the effects of a blood clot, and a blood clot has been induced somewhere up here, and that has blocked off blood supply to the rest of this tissue, and where it's yellow it has gone necrotic. So this is just purely down to cell death. So necrosis is a passive process compared to apoptosis, which is an active and deliberate form of cell death. So where we have necrotic cell changes, the key thing that we see is the disruption of the plasma membranes. So you get a disruption of the plasma membrane, and that allows the contents of the cytoplasm to spill out, and that leads to inflammation. And inflammation is not a good thing, especially when we're talking about wound repair, because if you've got inflammation, then you've got immune cells coming in, and that can often prevent it and result in scarring and long-term tissue damage. We also get disruption of intracellular organelles, so particularly a lot of proteases will spill out of the endoplasmic reticulum, as noted down here. And also if you get enzymes leaking out of the lysosomes, we'll cover lysosomes later on, but lysosomal enzymes, if they're released into the cytosol, they will degrade all the structures that they come into contact with, and the cell becomes autolyzed, which is why you then get these disrupted cells. In contrast, apoptosis is this normal physiological response to some sort of deliberate cell signaling, and the key changes that we see is nuclear condensation and cell shrinkage, and I'll show you those shortly. You get preservation of the organelles and the membrane, so you get an intact plasma membrane throughout the whole process. You then get the cell breaking up and pinching off and to form small membrane-bound fragments of cells. These can be phagocytosed, typically by macrophages, and they will clear up the debris left behind by an apoptotic cell, and as a result you get no inflammation due to apoptosis. So it's a very clean way of cells dying. The other thing that we see is DNA fragmentation. This is a biochemical hallmark of apoptosis, and I'll show you this in the coming lectures, where you get specific cutting of DNA between histone cores, and that will result in a classic signature of apoptosis. So all of these things happen, and we can exploit some of these changes to tell the difference between apoptosis and necrosis. And if you go to the PowerPoint version of the slides and click on this video, you'll see a link of all of the processes going on with an explanation of what is going on. That will be very relevant when we look at the apoptotic detection of cells in the lab next week. So typically when we get a cell undergoing apoptosis, it will go from being a rounded cell, this is just a lymphocyte, and you'll get this thing called a ruffled membrane or membrane blebbing. And what we find is that this membrane seems to shrink down, the cytoplasm shrinks, but then the membrane bulges out in these sort of outpinching bits of membrane, and these will eventually break off when the cell fragments. So we can see this down the microscope. When we look at cells undergoing apoptosis, you often see these big membranous bulges. But the membrane remains intact, it's not ruptured at all. And these slides illustrate what's going on. This is just a cartoon, hand-drawn diagram of what the cells look like down the microscope. So a normal cell, where we have got at point A, where we've got nucleus and cytoplasm visible. At part B, you see that the chromatin becomes much more granular. At C, you've got membrane blebbing. At D, we get what we call a crescent nucleus, and this is because during stages B and C, the nucleus is being degraded by DNAase enzymes, and that causes the chromatin to collapse onto the nuclear membrane, and then eventually at point E you get a condensed nucleus, and then you get nuclear fragmentation, and finally cellular fragmentation. And these points can be noted on this image here. This is a fluorescence microscopy image stained with the DNA binding dye DAPI, so we only see the nuclei. And at A we can see a normal cell with a large diffuse nucleus. We can't see the stages B, C and D in this image, but at E you can see a classic condensed nucleus, and there's another one there, and at stage F you've got nuclear fragmentation. So if we want to tell the difference between normal and apoptotic cells, we can just stain our cells with DAPI, and this DAPI stains the nucleus, and you can look at the nuclear morphology, and this is one of the gold standard methods of detecting apoptosis. And this little diagram shows, here's a normal cell, we've got an apoptotic trigger, we've got apoptotic cell shrinkage, so we get ion channels and water channels changing, particularly pumping out water out of the cell, or being released out of the cell. So we get cytoplasmic shrinkage, then we get DNA degradation, and then the cell fragments into the apoptotic bodies, which are then taken up by macrophages by phagocytosis. Now I've mentioned DNA fragmentation a few times, and this is a classic sign of apoptosis. Now, DNA fragmentation occurs because a DNAase becomes deliberately activated during apoptosis, and we'll find out about this in the second lecture that I'm going to put up today. So here is DNA, and DNA in the form of chromatin is wrapped around histone proteins, and histones are octamers, so there's 8 histone proteins with DNA wrapped around them, and then chromatin can be compacted into chromatin fibers such as this. So this is what chromatin is like in chromosomes. It's not just DNA, it is histone proteins. Now the important thing about histones is the DNA in between them are more available for DNA degradation, and this is where DNA gets cleaved during apoptosis. So here we can see these what we call nucleosomes, which are your histone cores, DNA wrapped around them. There's about 200 nucleotides from the start of where DNA finishes on one histone to the next. Now this means that if you cleave DNA somewhere between here and here, that was about 180 base pairs of DNA, and this is what we see, and we'll see later on in the module, that caspase activated DNAs, and this will crop up in the intrinsic apoptosis lecture, and it will crop up in the methods of analysis of apoptosis. This caspase activated DNAs becomes activated specifically in apoptotic cells. That caspase activated DNAs will cleave here and here, and it means that DNA in the cell that is apoptotic, the DNA is cut up into fragments which are multiples of about 180 base pairs. And we can run that DNA out on an agarose gel and visualize the fragments of DNA and see that this is classic for apoptosis. But we'll see that in much more detail later on, and I'll talk about CAD in the caspase section of the intrinsic apoptosis recording, which is coming up next. So cell death is tightly and intrinsically linked in with proliferation. If cells are receiving growth factors, they are being told to proliferate, and we've covered that in the cell cycle lecture. That generally means that a cell is in a position where there is plenty of nutrients, it's receiving growth factors, and that is driving proliferation. And growth factor signaling generally is anti-apoptotic, it's pro-survival. So mitogen signaling not only drives proliferation, it also switches off many cell death mechanisms and causes cells to survive. In contrast, cell starvation, and that's of mitogens, nutrients, can be of oxygen, can drive either apoptosis or can drive autophagy. Now, autophagy is a stage where the cell can enter a transient state where it can be devoid of nutrients and start to engulf its own cytoplasmic components for destruction. So this can be engulfing its own mitochondria and breaking them down and using those as a source of fuel if they are completely devoid of any nutrients. So this occurs in extreme starvation. As the cell is trying to avoid going into apoptosis, it can go into a transient phase called autophagy, and it can effectively, autophagy is self-eating in effect. So cells can be driven to autophagy by withdrawal of growth factors. If you withdraw them too far, then the cell will effectively give up and be encouraged to go down the pathway of apoptosis. So that's the basic explanation of apoptosis, necrosis and autophagy, and in the next lecture I'm going to explain how apoptotic signaling can be specifically induced in cells to drive them to deliberately undergo apoptosis or cell suicide.